is Christina Sartain. Thank you for joining me virtually today. I'm going to talk about my thesis in which I researched the effects of auditory predator cues on foraging patterns in the old field mouse, or Peromyscus polionotus. I did this project under the mentorship of Dr. Michelle Cawthorn and in fulfillment of the University Honors Program. Okay, so a little bit of background. I'm discussing predation risk. The ability to assess predation risk increases an animal's chance of survival and enhances fitness. This makes sense, of course, because if an animal is able to detect and avoid its predator, then it will live and pass on more genes and have more offspring, therefore having a greater fitness. Predator cues, or signals from a predator that the prey receive, help the prey animal to identify the predator and attempt to avoid predation. These can come in both direct and indirect forms and can be auditory, olfactory, or visual. Some examples of indirect forms would be moonlight exposure, temperature, precipitation, things like that. I looked at a study where tadpoles were exposed to the odor of a dragonfly larvae, and their responses were recorded, and what they found was that large amounts of odor elicited an anti-predator stress response, while small amounts did not. This could mean that in some species it is clear there is a reaction to predator cues, which could affect fitness levels and suggest that they do react to these cues, and it does affect their behavior. There's a bit of an arms race involved with predators and prey. Of course, with prey becoming better at detecting predator cues, and then predators being more able to avoid being detected. So it's a bit of a continual process that evolves and changes over time. The main idea here is that foraging behaviors are determined by a variety of factors and are strongly influenced by perceived predation risk. And to the best of my knowledge, the use of auditory cues in mice is only beginning to be explored. I only found one study on it, and it was only one of the variables they looked at. So I was interested to see if it did affect them or not. Really, the importance and significance of this topic here is that as behavior and feeding patterns are mapped out for any species, the relationships of coexistence within an ecological community are made clear and the habitat is better understood. So basically, the more we know about any species, the better we can protect their ecosystem and their numbers. And if an invasive species is introduced, or we want to do some farming or fertilizer, we are better able to protect them and reduce any harm that we might cause. And also, close relatives of Peromyscus polionotus are endangered. This includes the Alabama beach mouse, so better understanding of the Peromyscus family's behavior and foraging could help us in conserving that species that is rapidly declining. So my question became, as I researched this topic, was do direct predator cues in the form of predator calls affect foraging effort in Peromyscus polionotus as measured by the giving up density? The giving up density is a measurement of the amount that a mouse forages. So at what point do they consider the situation to be too risky and stop foraging? And previous studies are conflicting. I found one by Auroch that said only indirect cues affected foraging, so sunlight, temperature, things like that, and not direct cues from predators. But a 1972 study by Webster, his data supported the opposite. He found that direct cues did affect it. My hypothesis here was that direct predator cues in the form of auditory calls will significantly affect foraging effort as measured by the giving density. And my prediction was that P. polionotus will demonstrate a higher GUD in experimental trays emitting barn owl and coyote calls when compared to white noise or no noise as the control. Both of these predators are known to be found locally, with coyotes being more common than barn owls. Both are nocturnal predators in the old field mouse habitat and are active when they forage. We made sure that the area and meadow that we did this experiment in is a place that coyotes have been seen, barn owls have been seen, and things like that. So as far as methods, what we did is we located 5 active burrows, and we used 4 each trial with relatively equal vegetation cover near Meadow, Georgia. And why we did that to try and reduce the effect of indirect cues to focus on the direct cues. Each of them had 3 foraging trays, and we just used plastic Tupperware containers and an iPod emitting a predator call, whether that be coyote, barn owl, crickets, or no noise. And crickets and no noiser were both used as our controls. Each tray contained 3.1 grams of millet seed mixed into 2 cups of clean play sand. And we cut a 1 by 2 inch hole in both sides of each tray to provide entry to the mice, but to try and keep it small enough where deer or squirrels couldn't get in and mess up our data. And then the iPods were enclosed in a similar container. and we punched holes in it to protect from the rain but allow the calls to be as clearly heard as possible. The calls played on an alarm for 2 minutes every 4 hours over a 12-night period, and at the end of that period, data was collected for 2 to 3 nights periodically from November 2018 to March 2019, and then again in the winter of 2019 to 2020. Treatments were rotated among sites each trial. So we spread out the treatments, and the giving up density of the treatments versus the controls were compared for all trials combined. So here are some pictures to help you better get the idea. On the upper left here is an example of Peromyscus polionotus, the old field mouse. This is one caught by my mentor, Dr. Michelle Cawthorn. They're very cute and very small. And these are the creatures that we studied. And then on the bottom left here is an example of a burrow that I used with the active hole in the middle, the 3 equidistant trays around it, and then the iPod. And then on the right here are the sites that we used indicated by white X's. So for my results for trial 1, there were 17 sample nights, and there is a significant treatment effect here. So what we found was that the GUDs for the predator sites, so the trays that contain sounds for barn owl or coyote, were significantly higher than the GUDs for the crickets and silence sites. So it did show, or did suggest, that the mice did avoid the sounds of coyote and barn owl more than they did silence and crickets. What we didn't find, though, is that coyote and barn owl were not significantly different from each other, so it didn't appear that the mice were, quote unquote, more scared of one predator sound than the other, but there was an avoidance generally of a predator site versus a not. And there were 4 treatments. Each treatment had 45 replicates, except crickets, which had 42. So this is just a graph showing those results from trial 1, and as you can see, there is a significant difference between the barn owl and coyote sites versus crickets and silence, but there's not a significant difference in between the two, within the 2 groups. So we did get conflicting results, because in trial 2, where we had 10 sample nights, there was not a significant treatment effect. So what we found was that the treatments were not significantly different in the GUDs. They ranged in between 1 and 1.5, but there was no clear difference between the predator calls and the non-predator calls like there was in trial 1. There were, again, 4 treatments, of course, the silence, coyote, and crickets, and those had 30 replicates, but the barn owl had 29. So here we had a lower number of trials, but we did use a greater number of burrows, and we had our sites ranged over a greater distance, so it was interesting that there was not a significant treatment effect here. And here's just that graph showing trial 2, where there really isn't a significant difference between any of the 4 sites in terms of GUD. So unfortunately, my results were largely inconclusive. My prediction that GUD would be higher in experimental trays with predator calls than at trays with the non-predator calls is supported by trial 1, but not by trial 2. And this may suggest that mice do use indirect predator cues to dictate foraging patterns instead of direct. There are some notable differences between the 2 trials. Trial 1 was 3.5 months in the winter, trial 2 was only 1.5 months, and there was considerably more precipitation in trial 2. It was also considerably warmer in trial 2 than in trial 1, and more burrows were used in trial 2, which provided information on mice in varying areas. Trial 1 described a larger number of trials, but it was within a smaller area. All of these could have contributed to the difference in results, but we really don't know for sure. So in conclusion, further experimentation and more in-depth research is needed, of course, because our experiment had inconclusive results. So ideally, a field project of this nature should be conducted over the course of several consecutive seasons instead of just one, keeping all the indirect cues into account. And it should include an official biological survey of the area with confirmed and common sightings of all predator sounds used. And then I thought an accompanying lab experiment would really solidify the data, detailing mouse behavior when they're presented with a predator sound and no other cue around it, because you can really control for that in lab. My experiments and research have proved inconclusive on whether or not Peromyscus polionotus takes auditory predator cues into account when foraging, but further research and more meticulous methods could potentially find that they do. So that does conclude my presentation, and I wanted to thank Dr. Michelle Cawthorn for being my research mentor and advisor, for letting me use her land, her resources, her ideas. I really could not have done this project without her, and Destiny Martin for helping me set up and collect data during trial 2. She was a huge help. Thank you so much for listening.